When people of Black American descent take a consumer DNA test today, a significant number of them get results that do not match the story they grew up with. Not slightly different, genuinely, sometimes dramatically different in ways that no simple explanation covers. Some find ancestry percentages pointing toward regions of Africa they were never told their family came from. Some find Indigenous American markers their ancestry companies can barely categorize. Some find that the unassigned percentage sitting quietly at the bottom of their results, the one the company explains with language about statistical confidence thresholds, is larger than their entire reported European ancestry. And when they dig into what that unassigned category actually means, what they find is not a technical limitation that will be fixed next year. What they find is that the genetic history of Black America is layered, complex, and far older than the standard narrative was ever built to contain. This is not a story about replacing one identity with another. It is not a story about doubting or minimizing the African heritage that is absolutely central to Black American identity, culture, and history. The West African signal in Black American DNA is real, strong, documented, and present in the overwhelming majority of profiles that have ever been studied. Nobody serious disputes that. What this story is actually about is what else is in the data, what the reference panels are not yet built to surface clearly, what the haplogroup records show when researchers look carefully, and why so many Black Americans are getting results from testing companies that feel genuinely incomplete, even when those companies assure them the technology is working correctly. Let's start with the foundation. Most of what Black Americans are told about their genetic origins begins and ends with the Atlantic slave trade, specifically with the populations of West and West Central Africa, who made up the majority of people forcibly transported to North America between roughly 1619 and 1808. The genetic signal from those populations is absolutely there. Studies examining Black American mitochondrial DNA, which traces the unbroken maternal line backward through every generation, find L haplogroups at high frequencies. L1, L2, and L3 are the dominant mitochondrial lineages across West and Central Africa, and they show up strongly in Black American samples exactly as the historical record would predict. I-chromosome data tracing the unbroken paternal line shows E1b1a haplogroup, the primary West African male lineage, in approximately 65% of Black American men who test. The West African Foundation is not in question. It is real, it is well documented, and modern whole genome sequencing has only made it more precise and more detailed, connecting specific Black American lineages to specific ethnic communities in Sierra Leone, Nigeria, Senegal, Cameroon, Congo, and Angola at resolutions that simply were not possible a decade ago. But the remaining 35% of Black American male Y chromosomes, the ones not carrying the West African E1b1a signal, tell a more complicated story. A significant portion of that 35% carries European haplogroups, and the reason for that is not complicated or mysterious. The sexual violence embedded in slavery as an institution wrote itself directly into the paternal genetics of enslaved communities on an enormous scale. Somewhere between 35 and 40% of Black American men who take a Y chromosome test today carry a European paternal haplogroup, meaning their unbroken male line traces back to a European male ancestor rather than an African one. That is not ancestry the men who carry it chose. It is ancestry that the institution of slavery forced into their family lines through the rape of enslaved women by enslavers and overseers across generations. The DNA carries that history whether the Consumer Ancestry Report labels it clearly or not. What gets discussed far less is the portion of Y chromosome data that fits neither the West African nor the European category neatly. Research published on Black American male haplogroup distributions has found notable frequencies of R haplogroups in the sample population. R1b is usually attributed to European admixture through the slave trade, and in most cases, that attribution is probably correct. But R1b has its own documented presence in Central and West Africa that predates any European contact. In 2009, geneticists studying indigenous Cameroonian populations found R1b frequencies among Chadic-speaking communities that matched or exceeded the frequencies found in Western Europe. That discovery forced a significant re-examination of where R1b actually originated on the globe, and the debate is still ongoing. When a Black American man's Y chromosome comes back as R1b, his ancestry report typically attributes that result to European admixture. Whether that attribution is always accurate is a question the reference panels are not currently sophisticated enough to definitively answer. The reference panel problem is the single most important technical issue in Black American ancestry testing, and it is one that researchers have been documenting and criticizing in peer-reviewed literature for years. When a consumer testing company like Ancestry, DNA, or 23andMe tells you your ancestry breakdown, your DNA is not being compared against some neutral, universal database of all human genetic variation. It is being compared against a curated set of reference samples, real people whose DNA has been designated as representative of specific geographic populations. The accuracy of your result depends entirely on how complete, how diverse, and how representative those reference panels actually are for the populations in your ancestry. The African reference panels used in consumer testing have improved meaningfully over the past decade, and companies have made genuine efforts to expand them, but they remain disproportionately weighted toward populations most directly associated with the documented Atlantic slave trade, specifically the Yoruba, the Akan, the Fon, the Mandinka, and other West Coast populations. Central African populations are underrepresented. East African populations are underrepresented. North African populations are typically siloed into a separate category that does not connect cleanly to the African American experience, even though North African populations have been present in Atlantic world networks for centuries. A 2021 paper in Communications Biology identified this gap specifically in the context of Black American ancestry research, noting that there was a serious dearth of whole genome sequencing data for individuals with sub-Saharan African ancestry residing in the Americas, and publishing a new reference panel built specifically from 2,294 individuals to try to address it. What this gap means in practice is that when a Black American has ancestry tracing to a Central African population with limited reference panel representation, that ancestry does not disappear from their genome. It simply gets misattributed, assigned to the closest match available in the panel rather than the actual source population. Or it gets absorbed into the unassigned percentage that appears at the bottom of most consumer ancestry reports, labeled with language about confidence thresholds, described as ancestry. The algorithm could not confidently assign to any known region. Most companies do not report ancestry estimates below 1 or 2%. If someone has 3 or 4% ancestry from a population the reference panel cannot confidently identify, that ancestry may simply not appear in their report at all. Not because it is not there, because the system was not built to surface it. The Indigenous American marker question sits in a similar space of genuine complexity. A landmark 2014 study published in the American Journal of Human Genetics, examining the genetic ancestry of African Americans, Latinos, and European Americans across the United States, found that approximately 0.8% of Black American ancestry traces to Native American populations on a genome-wide basis. That is a small average number, but averages hide the variation across individual cases, and the distribution of that ancestry is not uniform. The study also found that mitochondrial haplogroups associated specifically with Indigenous American populations, specifically A2, B2, C1b, C1c, C1d, and D1, appear at meaningful frequencies in the Black American sample. These are haplogroups virtually absent in European populations, confirming that the Native American signal in Black American DNA is real and not a statistical artifact. What these numbers cannot easily tell you is the when and the how. Indigenous American ancestry in a Black American profile could trace to pre-colonial contact between free African and Indigenous communities, to the colonial period when Black and Indigenous communities throughout the Southeast had extensive direct contact, to the antebellum era when enslaved people and Indigenous people worked and lived in proximity on plantations, or to the post-emancipation period when Black and Native communities in the rural South mixed freely across generations without any of it necessarily being recorded in a surviving document. Distinguishing between those possibilities from a consumer ancestry result alone is genuinely difficult, and most consumer reports do not attempt to. The label Native American admixture appears in the result, and the rest is left to the individual to interpret. The Gullah Geechee communities of the South Carolina and Georgia Sea Islands represent one of the most thoroughly studied cases of Black American ancestry research, and what makes them especially valuable is that their documented history provides a partial check on what the DNA is actually showing. The Gullah Geechee descend primarily from enslaved Africans brought directly to the Sea Islands in the 18th century through a trade corridor that connected specifically to Sierra Leone and surrounding regions of West Africa. The geographic isolation of the Sea Islands meant that Gullah Geechee communities maintained extraordinarily high genetic and cultural continuity with their African origins. Their language, Gullah, preserves syntactic structures and vocabulary from West African languages that disappeared in every other part of the African American diaspora. Genetic studies of Gullah Geechee populations confirm exactly what the historical record says, with a very strong and specific Sierra Leonean and broader West African genetic signal. This is one part of Black American genetic history where the standard narrative maps almost perfectly onto the actual data. But even in the Gullah Geechee studies, researchers found layers that complicate the picture. Haplogroups appearing in the samples at frequencies that the West African reference panels did not fully predict. Indigenous American signals at rates higher than simple post-emancipation contact would be expected to produce. The populations most selected for genetic continuity with Africa still showed complexity. The documentary record alone could not explain what those extra layers represent, whether pre-colonial contact, whether colonial air mixing that plantation records never captured, whether post-emancipation community mixing in the rural southeast, is a question the data raises without conclusively answering. The Melungeon story is one of the most carefully documented examples of how free black ancestry in antebellum America produced communities whose genetic complexity confounded every racial category the colonial world tried to impose on them. The Melungeons are a mixed heritage community documented in the Appalachian region, primarily in the Cumberland Gap area spanning Virginia, Tennessee, and Kentucky, whose presence in American records reaches back to the earliest colonial encounters. European settlers who encountered them in the 1600s found communities already established, already speaking English or early English variants, already practicing settled agriculture. Nobody could determine where they came from. The Melungeons themselves had no single consistent origin story. Their appearance did not fit the racial categories colonists knew how to classify. Various explanations circulated over the centuries: Portuguese settlers, Turkish exiles, Cherokee descendants, free Africans who arrived before the formalization of the slave trade as a legal institution. In 2012, Jack Goins, a Melungeon descendant who had spent 40 years researching his community's history, led the core Melungeon DNA project to a published result in the Journal of Genetic Genealogy. What the study found was both clarifying and surprising. The majority of core Melungeon male lines carried haplogroup E1b1a, the sub-Saharan African male haplogroup. The maternal mitochondrial lines were predominantly European. The genetic evidence pointed to a founding population of free African men and white European women, producing children in colonial Appalachia, whose descendants spent the following 2 centuries identifying as Portuguese, as Cherokee, as anything other than what the racial laws of their era would have done to them had their African ancestry been officially recorded. Goins himself had expected his own Y chromosome to come back as European or Native American, based on his family's oral tradition. It came back as E1b1a, African. The Melungeons are not an isolated case. They are the most thoroughly studied example of a pattern that appears repeatedly across the Southeast and Mid-Atlantic. The red bones of Louisiana and Texas, the brass ankles of South Carolina, the issues of Virginia, the Lumbee of North Carolina, the Turks of South Carolina, who have maintained for 3 centuries that their ancestors were free Moors who arrived before the hardening of colonial racial categories. What these communities share is not a single common origin story. What they share is a genetic complexity, that the binary framework of the slave trade narrative was never built to contain free black ancestors who were never enslaved, indigenous ancestors who do not appear in plantation records, mixing that happened before the legal racial categories that would later define American life had been fully formalized. The Y chromosome data carries additional complexity that receives almost no mainstream attention despite being documented in published research. Approximately 40% of black American males who test their paternal Y chromosome find a European haplogroup, representing the direct genetic legacy of slavery's sexual violence. But among the remaining 60% who carry African paternal haplogroups, sub-haplogroup variation tells stories that go beyond simple West African origin. E1b1b, a related but distinct haplogroup associated with North African and East African populations, including the Berber peoples, across the Maghreb, and populations from the Horn of Africa, appears in black American samples at frequencies that require explanation beyond the standard Atlantic slave trade model. North African and East African populations were part of Atlantic world networks in ways that mainstream ancestry narratives rarely discuss. The genetic signal of that connectivity is present in black American genomes in documented, peer-reviewed literature. It simply does not appear in consumer ancestry reports because the reference panels were not built to surface it at the resolution required. The 5 Civilized Tribes story adds yet another layer to this complexity that is both genetically and legally significant, and it is still actively playing out in federal courts today. The Cherokee, Choctaw, Creek, Seminole, and Chickasaw nations each held enslaved black people before and during the Civil War. They also intermarried with black and mixed heritage populations across generations, including during the colonial period when the racial categories governing such relationships were still being legally constructed. By the 1890s, when the federal government compiled the Dawes rolls as the official census of tribal members, there were large numbers of people who were genetically and culturally both black and indigenous. The Dawes rolls separated them. Freedmen, the formerly enslaved, and their descendants were placed on different rolls from tribal citizens. That separation was not merely administrative. It carried direct legal force determining land rights, treaty benefits, and federal recognition for generations afterward. The genetic record underneath this legal and political history tells a different story than the Dawes rolls categories did. Studies of black communities in Oklahoma, Texas, and across the Gulf Coast have found elevated frequencies of Indigenous American haplogroups, particularly within the Q and C subclades that are specifically associated with deep multi-generational indigenous ancestry, rather than recent admixture from a single native ancestor a few generations back. The shared segment lengths in autosomal DNA, which researchers use to estimate how many generations ago two populations mixed, suggest in some cases that the indigenous ancestry in these communities is older and more extensive than a single great-grandparent would produce. The legal fight around Cherokee Nation citizenship for freedmen descendants reached the Supreme Court level before a settlement in 2017 restored citizenship rights that had been revoked. The DNA fight underneath that legal battle is about something different. It is about whether the identities the Dawes rolls imposed, black here, Indian there on paper with no legal mixing allowed, reflect anything like the actual genetic reality of the communities those roles were meant to categorize. The mitochondrial DNA complexity mirrors this. A study published in the Proceedings of the National Academy of Sciences examining African American mitochondrial haplotypes found that when black American samples were compared to African databases, more than 40% of haplotypes did not match any sequence in the database at all. More than half matched sequences shared across multiple African ethnic groups, meaning precise regional attribution was impossible with the panel available. Fewer than 10% matched a single specific ethnic group. The genetic record of black American maternal ancestry is simply more diverse, more globally distributed, and more resistant to precise regional attribution than the standard consumer report category of West African or Nigerian suggests. None of this complexity reduces the centrality of the West African heritage that defines black American culture, music, language, religion, art, and lived identity in America. The African foundation of black American identity is not threatened by genetic complexity. It is deepened by it, because what the genetic record shows when you look carefully is a community whose origins stretch deeper into human history, connect across more of the globe, and carry more layers of survival, mixing, resistance, and endurance than any single origin narrative could hold. Slavery as an institution was a system of erasure on an industrial scale. It erased names, languages, family structures, religions, and oral histories across generations. The genetic record is one of the few archives that erasure could not fully reach. The oral traditions that survived within black American families across the Deep South are part of this evidence too, even though they have historically not been treated as such by formal genealogical and genetic research institutions. Across black American communities in Georgia, Mississippi, Alabama, the Carolinas, and Louisiana, there are family stories passed down for generations about ancestors who were not brought from Africa on ships, ancestors who were already on this continent, ancestors who were native or mixed or came from somewhere the family could not exactly name but knew was not the west coast of Africa. These stories were sometimes suppressed, not always by external forces, but sometimes deliberately within communities themselves, because claiming indigenous or pre-colonial ancestry complicated legal and political categories that were important to maintain during periods when access to historically black institutions, political coalitions, and community protections depended on accepting a unified origin narrative. The complexity was not always safe. It was not always strategically useful. So it went underground, survived in what grandmothers told children privately, in family naming patterns, in foodways and practices that did not match any West African tradition documented in the standard literature. Now, those oral traditions are meeting genetic results that validate what the paperwork could never prove, and the experience for individuals is often described as disorienting in a specific way. It is not just surprise. It is the feeling of holding a document that confirms something you always half believed but had no official evidence for, combined with the uncomfortable recognition that the authorized story you were given, the one that told you your history began on a slave ship, was not wrong exactly, but was not complete either. The complexity inside it is not a problem with the data. It is the data telling the truth about a history that was never as simple as the authorized version claimed. The testing technology is improving faster than the interpretation frameworks can keep up with. Within a decade, the unassigned percentages in black American ancestry results will likely begin resolving into something more specific, more regional, more named. When they do, the portrait that emerges will almost certainly be more ancient, more globally connected, and more layered than the story the consumer ancestry industry currently has the reference panels to tell. The African American DNA Roots Project, run through Family Tree DNA, has been working at precisely this level of resolution for years. Rather than the continental-level attribution that consumer tests provide, this project collects and analyzes Y chromosome and mitochondrial DNA samples from black Americans specifically to identify which African ethnic communities the transatlantic slave trade drew from at the level of individual groups rather than broad regional categories. The project has made significant documented progress connecting specific Black American lineages to communities in Sierra Leone, Senegal, Nigeria, Cameroon, the Congo Basin, and Angola in ways that a standard consumer ancestry percentage cannot approach. The difference between knowing you have Nigerian ancestry and knowing your maternal line traces specifically to the Igbo community of southeastern Nigeria, or your paternal line to the Tikar people of northwestern Cameroon, is the difference between a blurry outline and an actual face. That level of resolution is now achievable for some lineages, and the work of projects like this one is gradually making it achievable for more. What all of this points toward is a simple conclusion that takes time and evidence to fully absorb. The genetic history of Black America is one of the most complex, most layered, and most globally connected stories in all of human population genetics. It begins in Africa, deeply and unmistakably in the haplogroups and genomic patterns that the transatlantic slave trade stamped into the population. But it does not end there. It runs through free Black communities that existed before the legal consolidation of slavery. It runs through indigenous North American populations whose interaction with Black and mixed heritage people began before the first slave ship and continued for centuries after the last. It runs through Appalachian mountain communities that spent 200 years disguising their African origins behind Portuguese and Cherokee identities to survive in a legal world that would have enslaved them otherwise. It runs through Sea Island communities that kept the genetic and cultural memory of Sierra Leone alive in their language and their DNA across 12 generations. And it runs through percentages marked unassigned in consumer reports. Percentages that are not errors, not noise, not limitations to be embarrassed about, but information waiting for the reference panels to finally become sophisticated enough to read it clearly. The story of Black American DNA has never been only what anyone officially authorized. It has always been this complex. The technology is finally starting to catch up with the truth. And as whole genome sequencing becomes affordable enough for individual researchers and genealogy projects to run on population samples without institutional budgets, and as the ancient DNA field extracts usable genetic material from remains that were previously considered too degraded, the gaps in every reference panel are closing, slowly and unevenly, but closing. The algorithms that compare modern samples to ancient genomes improve every cycle. Each improvement means more of those unassigned percentages begin to resolve into something nameable. Each improvement means another family's oral tradition, the one nobody outside the household ever believed, gets a genetic document attached to it. The archive inside the DNA of Black America is deep. Researchers and genealogists working at its edges are nowhere close to done exploring it. But with every passing year, the picture gets a little clearer, a little more specific, and considerably more honest than what any single standard origin narrative was ever designed to hold.